morning. Welcome to Biology. I'm Paul Suchecki, and this is my classroom. It's good to see everybody's smiling faces again today. Today's lesson is on nucleic acids, DNA, and RNA. The incredible diversity of living things on Earth never ceases to amaze me. And the incredible part is that all species on Earth share the same genetic code. DNA is the molecule that stores that code. But unlike computer code, no human invented it. Nature invented it. The genetic code is a universal code that is expressed in endlessly diverse ways to overcome the complexities of staying alive. It seems reasonable that similar species with similar traits share similar genes. They must have similar DNA. For example, there are genes in our DNA that control how our liver works. Well, you have a liver and I have a liver. So the genes that you have and the genes that I have that control the liver are probably real similar. As a matter of fact, your DNA and my DNA are probably about 99.9% similar. What that means is, in terms of DNA, we're really only different by 0.1%. There's a lot more that makes us similar than makes us different. Well, a dog also has a liver, and their liver performs basically the same function as our liver. There are genes in a dog that control how its liver functions. It seems reasonable that the genes in a dog that control the liver and the genes in a human that control the liver are probably similar. Turns out that dogs and humans share about 84% similarity in DNA. After all, humans and dogs are both in the animal kingdom. But what about species that don't seem to be similar at all, like humans and a banana? Turns out that about 50% of our DNA is similar to a banana. Crazy. I guess we have more in common with bananas than you think. DNA has been passing genetic information from parents to offspring for countless generations. The DNA molecule must be remarkably stable to pass this genetic information on accurately, and yet have enough mutability to allow species to change and adapt to their environment over time. It's been less than a century since science began revealing the secrets of DNA, and now society stands on the brink of some of the most amazing breakthroughs in medicine and science since the discovery of the double helix. Nucleic acids, like DNA, are the genetic material of life. DNA stands for deoxyribonucleic acid. DNA stores and transfers genetic information. DNA also controls the production of protein. DNA is a double helix, found in the nucleus of a cell. The DNA double helix is composed of two strands of DNA held together in the middle and twisted. It's kind of like a twisted ladder or a spiral staircase. RNA is DNA's cousin. RNA stands for ribonucleic acid. RNA is like DNA's helper. RNA helps in the production of protein. RNA is not a double helix. RNA is just a single strand. RNA can be found in the nucleus or in the cytoplasm. Nucleic acids are made of smaller building blocks called nucleotides. A nucleotide is composed of three parts: a phosphate, a sugar, and a nitrogen base. There's nothing particularly interesting about the phosphate part. It's the same in both DNA and RNA, and it makes up part of what we call the sugar-phosphate backbone that holds the molecule together. The sugar part of the nucleotide is always a 5-carbon sugar. If it's an RNA nucleotide, then it's ribose sugar. The formula for ribose is C5H10O5. But if it's a DNA nucleotide, then it's deoxyribose sugar. The formula for deoxyribose sugar is C5H10O4. Ribose, C5H10O5. Deoxyribose, C5H10O4. Get it? Deoxyribose has one less oxygen atom than ribose. But that's just the sugar part. The more interesting part of the nucleotide is the nitrogen base. That's where the code is located. The nitrogen base could be one of 5 things. It could be a molecule of adenine. Let's just call it A. It could be guanine. Let's just call it G. It could be thymine. Let's just call it T. It could be cytosine. We'll just call it C. Or it could be uracil. We'll just call it U. DNA uses the nitrogen bases A, T, G, and C. RNA uses the nitrogen bases A, U, G, and C. The only difference is the T and the U. But in DNA, these 4 letters, these 4 nitrogen bases, make up the genetic code. If you're familiar with computer code, then you know that computers use just 2 symbols, a 1 and a 0, to store all kinds of information. How much information can a computer store with just two symbols, a 1 and a 0? Yeah, lots. Well, DNA uses 4 symbols to store information. But the information's not stored electronically, it's stored chemically. In DNA, these 4 chemicals are used to store information in units called genes. A gene is a segment of DNA that holds the information for one protein. For every protein in the body, there's a gene somewhere in your DNA that codes for it. Some genes can actually code for more than one protein. It's the sequence of nitrogen bases in that gene that determines how the protein is constructed. The way your proteins are constructed determine whether you're going to be a bunny rabbit, a human, or a carrot. Here is a DNA nucleotide. Here's another one, and another one, and another one, and another one, and another one. DNA is made of a long chain of DNA nucleotides. But DNA is a double helix, so it's got 2 sides. Here's the other side. These 2 strands are held together in the middle by weak hydrogen bonds that pull apart easily and are reformed easily. Notice that the 2 strands run in opposite directions. One side runs in the 5' to 3' direction. The other side runs in the opposite direction. That's no accident. The DNA molecule is said to be anti-parallel. The 2 strands are parallel, but they run in opposite directions. Notice that wherever you see an A nucleotide, it's always matched with a T. And wherever you see a C nucleotide, it's always matched with a G. A always matches with T, and G always matches with C. This is just a small segment of DNA. But in fact, a DNA molecule could be billions of nucleotides long. In human cells, there are about 3 billion nucleotides that make up our genome. It's the sequence of these nucleotides that spell out the genetic code. It's what makes your eyes blue or your hair red. No human invented the genetic code, and we're just beginners at reading it. Scientists don't know what all those letters mean yet, but they're working on it. In fact, every cell in the body has all of the DNA for the whole body. In other words, a skin cell has all the genetic information about how to be a skin cell, but it also holds genetic information about how to be any kind of cell. The genes for skin cell are turned on, and all the other genes are turned off. Likewise, a liver cell has all the genes on how to be a liver cell, but it also has all the genes for the whole body. In a liver cell, only the liver genes are turned on, the other genes are turned off. Scientists would like to know how to turn some genes on and some genes off. I don't think they can do that yet. But if they could potentially turn a skin cell into a liver cell. Now that would be something. DNA makes an exact copy of itself through a process called DNA replication. The DNA replicates every time the cell divides. That way, each daughter cell gets a full set of genetic instructions. Replication starts at the origin of replication. An enzyme called helicase causes the DNA double helix to untwist and pull apart, forming a replication fork. This exposes the nitrogen bases. Free-floating nucleotides found in the nucleoplasm match up with the unpaired bases. An enzyme called DNA polymerase bonds them together. A matches with T, and G matches with C. This process continues through the entire DNA molecule until 2 identical pieces of DNA are formed. There's just one small hitch. The enzymes that control replication can only replicate in one direction. DNA can only replicate in the 5' to 3' direction, and the 2 strands are anti-parallel. One strand, called the leading strand, has it easy. The leading strand replicates continuously toward the replication fork, nucleotide after nucleotide after nucleotide, until it reaches the end. The lagging strand replicates discontinuously away from the replication fork in little fragments called Okazaki fragments. And then these fragments are bonded together by an enzyme called ligase to form a continuous chain. In the end, there should be 2 identical pieces of DNA. I say should because sometimes cells make mistakes. Cells are busy places, and replication happens very fast. Sometimes the cell substitutes a wrong letter. Sometimes it skips a letter. Sometimes it adds an extra letter. These are all mistakes. A mistake or a misspelling in the DNA is called a mutation. and mutations are random events. A mutation could cause a handicap or even death. Potentially, a mutation could be a good thing and give an organism an advantage and help it survive better. This is what drives evolution. Or a mutation could be silent and have no consequence at all. We all contain mutations. Any organism that contains a mutation is called a mutant. Yeah, we're all mutants. It's the mutations that make us all different. If it weren't for the mutations, we would all be the same. Sometimes mutations happen for no reason. Cells just make mistakes. You gotta cut the cell some slack though. It has to copy 3 billion bits of information in about 20 minutes, without a mistake. That's not always easy, even under the best circumstances. But if we start throwing distractions at the cell, like radiation, or chemicals, or drugs, or other substances that can cause mutations, it can increase the chances that a cell could make a mistake. Any substance or other environmental factor that can cause genetic mutations is called a mutagen. There are mutagens everywhere, so be careful. DNA is responsible for protein synthesis. Protein synthesis is the production of protein. Proteins are long chains of amino acids, and there are thousands of proteins in your body, made from just 23 different amino acids. Think of amino acids like letters and proteins like words. There are 26 letters in the alphabet. How many words can you make with just 26 letters? Yeah, look in a dictionary. That's how many words. Well, how many proteins can the body make from just 23 amino acids? Thousands. What makes one word different from another word is just the sequence of letters. You could take a word and rearrange the letters and come up with a whole different word. It's like protein. If we arrange the amino acids in one particular sequence, we get one protein. But if we put the amino acids in a different sequence, we get a different protein. It's the DNA that determines the sequence of amino acids. A gene is a segment of DNA that controls the production of a protein. Protein synthesis occurs in 2 steps: transcription and translation. They're in alphabetical order. During transcription, the information for a protein that's on one gene is copied from DNA onto messenger RNA, or just mRNA for short. During transcription, the DNA opens up at the gene to be transcribed, exposing both sides of the DNA molecule. However, only one strand is copied. The strand that's copied is called the sense strand, and the strand that's not copied is called the nonsense or antisense strand. For any given gene, scientists aren't sure which side is the sense and which side is the nonsense side. But the cell knows, and that's what's important. Free-floating RNA nucleotides match up with the nucleotides on the DNA sense strand. The DNA merely acts as a template for the building of the mRNA. A matches with T and G matches with C, but in the case of RNA, U matches with A. Anywhere that you want to put a T, put a U instead. When all the RNA nucleotides are joined, the mRNA strand leaves the DNA template. Transcription is done, and the mRNA leaves the nucleus and goes out into the cytoplasm. This copy of mRNA dictates the sequence of amino acids for a specific protein. The second step of protein synthesis is called translation. During translation, a ribosome reads the mRNA 3 letters at a time, and the corresponding amino acids are placed in the correct sequence to form a protein. Each 3-letter sequence on the mRNA is called a codon, and each codon represents a specific amino acid. This is a copy of the genetic code. As mentioned earlier, the genetic code is universal, and every species uses the exact same genetic code. This is a piece of mRNA. A ribosome is going to read this 3 letters at a time and place the correct amino acids in the correct order. Find the first codon. The first codon says AUG. Look at the genetic code. Find AUG on the genetic code. It says start. AUG doesn't really represent an amino acid. It's what we call a start codon. It tells the ribosome, start reading here. It's kind of like the capital letter at the beginning of a sentence that tells the reader this is the beginning of the sentence. The start codon does the same thing. Find the next codon. In this case, it's UCU. Find UCU on the genetic code. Which amino acid is represented by the codon UCU? It says serine. Serine is an amino acid. A molecule called transfer RNA carries the amino acid to the codon UCU and places serine in the correct place. Now we're cooking. Find the next codon. AGG. Find AGG on the genetic code. Which amino acid is represented by the codon AGG? I see arginine. So a transfer RNA molecule is going to carry the amino acid arginine to the codon AGG and place it in the correct spot. And the ribosome joins the serine and arginine with a peptide bond. Now we have a dipeptide, two amino acids joined together. Let's move on to the next codon. The next codon is CUG. Find CUG on the genetic code. Which amino acid is represented by the codon CUG? It says leucine. So a transfer RNA molecule carries the amino acid leucine to the codon CUG. The ribosome bonds the leucine to the arginine. Now we've got three amino acids joined together. What's the next codon? The next codon reads UGA. Find UGA on the genetic code. It says stop. UGA is what we call a stop codon. It's not the only stop codon, but a stop codon is like the period at the end of a sentence. It tells the ribosome that's the end. Now, this is just a simple imaginary protein that I made up. It has only 3 amino acids. A typical protein could be up to 100 or more amino acids. What if the cell makes a mistake and accidentally puts the wrong letter right here? Well, that's a mutation. It's a misspelling. The original codon read AGG, but now it reads AAG. That's a mistake. Find AAG on the genetic code. That's different. That says lysine. So instead of arginine, the cell's going to put lysine there. That's a mistake. It's not the same protein anymore, and it probably won't function the way it's supposed to. If that's an important protein that you can't live without, big trouble. But if it's not a terribly important protein, you might just get by. So here, let me change that back to AGG instead of AAG. Now, what if that same mutation occurs just one letter over? So now, let's change it from AGG to AGA. Find AGA on the genetic code. AGA represents the amino acid arginine. Well, that's not different. It's still considered a mutation because it changed the DNA, but it didn't change the amino acid sequence, so it didn't change the protein. Since the protein wasn't changed, we call it a silent mutation, and it basically makes no difference at all. Protein synthesis is a complex process, and to be honest, I left out some of the nitty-gritty details. If I included all the nitty-gritty details of protein synthesis, it would be overwhelming, and then we would get lost. And you know me, I don't want anybody to get lost, at least not on my watch. Scientists don't know everything about DNA yet, but they make new discoveries all the time. It seems like every day new discoveries are made. I suppose the ultimate goal would be to determine what all of those letters mean. Where's the gene that makes your eyes blue? Where's the gene that makes your hair red? As scientists begin to read the genetic code, the hope is to cure all kinds of horrible genetic diseases, to unlock the secrets of Alzheimer's disease and diabetes and cancer, and hopefully find cures for these things, to produce drought-resistant or insect-resistant food crops. The potential is really endless. Scientists think that this technology could be used to bring back extinct species, like the dodo bird, or even dinosaurs. It might be cool to bring back a dodo bird, or even a woolly mammoth, but I'm not sure bringing back a T-Rex is such a good idea. I saw that movie. It didn't turn out so good. But like they say, sometimes you get the T-Rex, and sometimes the T-Rex gets you. But science is a tool, and like any tool, it can be misused. Humans need to think carefully about how we're going to use this knowledge. I guess we'll just have to leave that up to the politicians to figure out. But for some reason, that's just not comforting. Well, hey, I'm going to stop there and leave it there for today. I hope you had fun. I hope you learned something. Till next time, don't get lost. Have a great day, and we'll see you then.